Thank you for standing by and welcome to the Imugene Investor Webinar and Q&A today, discussing the FDA IND Clearance for onCARlytics. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 10 minutes before we get into some Q&A. If you'd like to submit a live question, please do so using the Q&A panel within Zoom. On the webinar for Imugene today, we have the CEO and Managing Director, Leslie Chong, and joining her is Dr. Saul Priceman, who's the co-inventor of onCARlytics and a member of Imugene's Scientific Advisory Board. To start off, I'll hand it over to Leslie. Please go ahead.
Hello, everyone. I am extremely excited to announce that we were able to get the FDA IND cleared, actually earlier than we had anticipated, and the team has been working on this, as you can imagine, for a good year, getting the IND enabling studies together, working closely with Dr. Saul Priceman and Dr. Anthony Park, and as well as Professor YUMAN Fong. I'm extremely delighted. This is the first in class, and we hope that it's the best in class. It's the first time an oncolytic viral therapy has a transgene that expresses a protein that interacts or combines with a CAR T or a CD19 directed therapeutics that we can hopefully obliterate solid tumors as we know it. So I'm happy to have Saul present to you all the background, and then we can entertain your questions. So thanks so much for joining. Next slide, please. Our disclaimer, next slide. So Saul doesn't need much introduction from me. I think he's been pretty prolific. Some of you have already met Dr. Saul Priceman when he was out here in 2022. We hope to get him out here very soon so that he could talk to everyone about this particular asset and how we're going to put it in the clinic. So Saul and I have become fastly best friends. I think we became best friends when we met. So without further ado, I'm gonna switch this over to him and have him explain onCARlytics. Next slide, please.
That was very sweet, Leslie. So it's a pleasure to be here. This is a very exciting time, extremely exciting, and faster than we could have ever have imagined. And this technology really stems from the basis with an oncolytic virus, and I won't go through too much detail, but oncolytic viruses are extremely interesting living creatures that have been trained to infect tumor cells specifically. They can infect them in lice and kill those tumor cells, and in doing so, they actually replicate and infect surrounding cells. And the most interesting thing, and very directly related to the combination I'll describe in a few minutes, is that they can carry genes that we engineer into them that are delivered specifically to the tumor. And interestingly, and as YUMAN would describe it, who's the real expert in this part of the combination, the oncolytic virus, is that for many years, people have been sort of afraid of using these oncolytic viruses, and it has hindered the field. We've only seen one FDA approval to date in the US using an oncolytic virus called THERAPIES, and it's used for melanoma. And if you go to the next slide, please. The idea that YUMAN had several years ago is that he could make a chimeric version of an oncolytic virus with far greater potency, meaning it could infect tumor cells at levels that had never been described before, and it could be uncoupled from infecting and killing normal cells and impacting safety of this. And in doing so, he batched several viruses, 9 in total, together to make a chimeric oncolytic virus, tested it against the NCI cell line 60, which really reflects all the solid tumors that could be used to treat with this oncolytic virus, and showed that it was highly potent. Of the hundreds that he tested, he picked out one that he called CF33. And in high throughput screening, he then took it to animals and showed dramatic safety and anti-tumor activity, and termed this HOPE oncolytic virus. This was actually licensed to Imugene several years ago. And if you go to the next slide, really what has happened is a number of clinical trials have have come about. I'm going to kick it back over to Leslie to tell you how exciting the trials that are underway are, and then I'll come back and describe the combinatorial therapy.
Thank you. So we already have a well-advanced study that we call CHECKvacc. This is the CF33 backbone, human's invention of the oncolytic virus, with the transgene of PD-L1. So it gives a direct payload to the tumors. And this is in triple-negative breast cancer, which is the deadliest diagnosis of a breast cancer. These patients have come on after several lines of failing other lines of therapy. So they're well treated, and we're in cohort 3. So we're just rounding out cohort 3. We will interrogate probably a higher dose because we'd like to obtain what's called a recommended phase 2 dose from the study, and then hopefully move on to another study. Next slide, please. So this is the parental virus. So this is the onCARlytic virus. The oncolytic virus in the most powerful form. As you know, the study started in May of 2022, and already we have cohort 3 in intratumoral dose as well as intravenous dose, and in combination with pembrolizumab. We're very excited because this study gives us a lot of initial data to move forward in our onCARlytics. This is, again, in multiple types of solid tumors, and we have exactly that. We have testicular, we have ovarian, we have breast cancer, we have melanoma. So it's a hodgepodge of various different solid tumors because, again, we think that the parental virus CF33 multiplies in multiple types of tumor types. So we're proving this in our studies now. Next slide, please. And I'll switch it back to Saul to explain onCARlytics.
Yeah, so I'll preface this with stating 2 facts that went into this. One is that Imugene had now created this oncolytic virus that had preclinically been shown to infect very specifically tumor cells, not normal cells, showed dramatic safety and really potent and effective anti-tumor activity across a number of solid tumors. And in the field of CAR T-cell therapies, chimeric antigen receptor T-cell therapies, which have taken by storm hematologic malignancies, liquid cancers like leukemia and lymphoma, targeting a protein called CD19, really was the introduction to me and my lab developing CAR T-cells for solid tumors. But what we've come to realize is nothing is quite like a hematologic malignancies. We've had really effective therapies targeting CD19 with CD19 CAR T-cell therapies, and nothing exists like that for solid tumors, whether it's prostate cancer, breast cancer, pancreatic cancer, and so on and so on. And in one meeting with several of us, what spawned out of that was the idea, well, if an oncolytic virus can truly selectively target a solid tumor and can carry with it a payload, why couldn't we recreate a liquid cancer in a solid tumor, bring CD19 to a solid tumor, whatever that solid tumor is, and then we can target it with the most effective CAR T-cell that we know, which is a CD19 CAR T-cell. And that's exactly what we did. And what happened is I punted this over to Anthony Park in my lab, and the rest really is history. The idea was we were going to infect cells with CF33 carrying CD19, infect tumor cells, breast cancer cells, for instance, and then we could treat those cells with a CD19 directed molecule. And that was the basis for onCARlytics. Next slide, please. And so this is just the summary of what we showed, which was since published in a very prestigious journal, essentially showing that the onCARlytic could infect any tumor cell, as you can see on the left: pancreas, prostate, ovarian, head and neck, glioma, and a number of others. And on the right, with that virus in combination with a CD19 CAR T-cell that you wouldn't expect to have any activity in a solid tumor, we were wiping those tumors away. Regression that we've not seen before. And we had data in this to demonstrate that there was safety of this approach, at least in preclinical studies. And if you go to the next slide, please. We published this in a very prestigious journal. We actually got the cover. And really the idea is that we've now, we think, generated a universal immunotherapy approach that can treat any patient with cancer, which is unlike what we've typically done in treating specifically breast cancer, specifically prostate cancer. This type of approach, we think, we can apply to anyone with cancer, irrespective of their specific disease. On the next slide, please. We've actually expanded this in collaborations with a number of companies that are developing CD19 targeting agents and different cellular backbones that we can combine with onCARlytics. And we presented this in 3 independent posters at the Society for Immunotherapy of Cancer in 2022. On the left, collaborating with a company called Celularity, who's making what's called allogeneic T-cell therapies, where they can make a bank of cell therapy from a healthy donor and treat hundreds of patients with cancer. And in the middle, using a very novel approach with a CD19 CAR T-cell in collaboration with Eureka Therapeutics. And on the right, using a non-T-cell type of approach, but engineering that cell with CD19. So all in hopes of applying multiple combinations for patients with cancer. And on the next slide, please. The approach we've also combined with non-CAR T cells and expanding this into a fully off-the-shelf approach where we can take drugs from one freezer, another drug from another freezer, and combine them and treat patients, as opposed to making CAR T cells, which is usually a patient-specific therapy. And we presented this recently at SITC as well, and another conference called ASGCT. And so this combination is using a bispecific T-cell engager that brings CD19 towards a T-cell and effectively has the same outcome. And I won't go through the detail here. There's a lot of data, but essentially causing regression of solid tumors using the combination with onCARlytics. And on the next slide, please. We've also presented additional data, really potent anti-tumor data using Eureka's CD19 CAR T cell as well, essentially expanding what we know now to be the combination, but being able to use multiple therapeutic modalities. And I think on the next slide that this is it. Just a nice illustration showing the universal modality here with our onCARlytics platform, and we're really excited. I mean, this is a milestone that we've reached, and the collaboration with Imugene has been just stunning. We're really excited about this. Thank you.
I think that's the end of our presentation. If we wanted to open it up for questions, please.
Thanks, Leslie, and thanks to Saul as well for that. As you've mentioned, we'll move on to the Q&A session. If anyone would like to submit a question, just a reminder, you can do so using the Q&A panel within Zoom. I'll cover a first that came through via email, though. First one says, I read that the onCARlytics trial will run in the United States. Just wondering if any additional trial sites will open in Australia, either now or in the future.
Sure. We're starting in the US because we've got some prominent sites that will be conducting the study initially, including, obviously the City of Hope, where they have a lot of experience with our CF33. So we want to take sites that have an understanding of not only an oncolytic virus, but also CAR T or CD19 directed therapies like Blincyto before we move into the sites.
Thank you. The next question is, what's the regulatory approval pathway look like to get onCARlytics to market should clinical trials prove it is effective, as we all hope?
So the general rule of thumb in the past had been that an asset or a technology or a drug would have to be moved into the clinic for roughly about 15 years, and that's actually the average. With the onset of immunotherapies, that's completely move the dial, and the FDA has to be a little bit more innovative in terms of how they look at drug development and drug approval. So we are hoping that we fall into that space where it's all about what happens in the clinical trials and the responses that we see in patients. We hope to move the dial in terms of that timeline. But again, likely we'll move into a phase 1 and have some clear signals, clear response, so that we can move right into a phase 2 registrational stage. Now, as to how many patients, that timeline, that's unclear, but likely with something like this, we will move as quickly as we can, because we'd like to have patients to be able to access this drug.
Thank you. I'm going to turn this into a two-part question. There's quite a few asking about when you expect the first patient to be dosed, and then in line with that, also, how long do you think it will take approximately to dose the first cohort?
I think we're going to get first patient in as early as this year, and depending on, what we're seeing in the MASS study, our monotherapy as well as combination, people are lining up their patients for these studies, so we'll likely move quite quickly. As to that time and the staggering that FDA puts on us, that will likely impede some of the enrollment timelines. However, I'm hoping that in 2024 we can get patients on, and maybe even the first few cohorts.
Thank you. A question for Saul. Dr. Priceman, would you please give some insight of what is the mechanism of CF33 only replicating in cancer cells and not normal cells? Is there an explanation at the molecular level? Thank you.
Yeah, so, viruses typically use specific machinery to get into cells. This vaccinia virus uses receptors on the surface of tumor cells that are not found or found at very low levels on normal cells. So even the infectability of the virus is improved in tumor cells, and we've seen that time and time again in the laboratory. The other aspect is that it's thymidine kinase dependent, which is a host machinery found highly expressed in tumor cells. And so overall, the bias toward tumor cells we've demonstrated using this virus.
Thank you, Saul. The next question I have is, how does onCARlytics viral potency compare to the parental vaccinia virus? It looks like it only has one transgene payload, just like vaccinia, compared to the 2 transgenes in CHECKvacc.
What we've seen to date is that the more payload, potentially the less infectious the virus might be. And one can overcome that with dosing the virus at higher viral loads. But that is right. This one has one payload like vaccinia, and should be quite potent.
Thank you. The next question is, if the OV/combination therapy is successful in killing tumours in humans, what happens to the virus in the body after cancer is eradicated?
In tumors, what we think is that there's immune tolerance to some extent. The virus should persist longer inside tumors than it would systemically. If there's no tumor, we believe the virus would get cleared like other viruses would be.
Thanks, Saul. The next question I think is back to you, Leslie. When you say you hope to move the dial, does that mean potentially applying for breakthrough therapy if the data is as expected?
Of course, breakthrough therapy, fast track, all of those regulatory terms and certificates that gets you to literally move faster.
Great, thank you. The next question is there any risk or condition under which this virus attacks normal cells? What would be the effect if that happened?
I'll let Leslie speak on any of the safety data to date. But the way that we're approaching the combination clinically is that we're giving the time for the virus to really infect tumor cells specifically prior to giving the combination. And at least in our preclinical studies, there was safety demonstrated, and we expect that we'll see some similar safety clinically.
Did you have anything to add there, Leslie?
Would you repeat the question again? I was looking at the questions coming in.
No problem. It was just around, is there any risk or condition under which the virus attacks normal cells? What would be the effect if it happens?
Right. So we have not seen this in our current mass study or the CHECKvacc study, where is replicating in healthy cells, which is exactly what we saw preclinically. We anticipate that the onCARlytics, our CD19 or onCAR19, will behave the same. However, we have put some very, very careful perimeters around the protocol and the conduct of the study, such that we are not harming patients or their healthy cells.
Great, thank you. And final question I have is, can the CD19 oncolytic virus be used in conjunction with targeted molecular therapy? For example, forgive my pronunciation, larotrectinib targeting NTRK fusion for pancreatic cancer.
You know, we're exploring preclinically any number of combinations. We think that this combination and its synergistic activity may be sufficient, but we are exploring in the lab other combinations to even further improve the activity. And specifically with drugs that are applied to specific cancers like pancreatic cancer, we would take those into consideration as we expand into those diseases with the combination.
Thank you. And the absolutely final one, for other cancer drugs like Keytruda, PD-L1 is an important marker for the usage of the drug. In CF33's case, is there any relevant biomarker to look for?
Well, I think that's the beauty of onCARlytics, that it wipes away, or the mosaic nature of solid tumors, this very heterogeneity. We're actually obliterating that. So we're making everything into a CD19 target. That's the gorgeous nature of this drug.
Great. Well, that concludes the Q&A. Thank you, Saul, again, and thanks, Leslie. I'll throw it back to you, Leslie, just to provide a closing comment.
Look, thank you so much for joining us, and thank you, Saul Priceman, for being my partner in crime today. We're incredibly, exuberantly excited about this going into the clinic and getting FDA IND without any sort of issues. It's been a beautiful ride. It's years in the making to get this into the clinic, and we just can't wait for patients and cancer drug landscape.
Great. Thanks, Leslie. Thank you again, Saul, and thanks to everyone for joining today. That concludes the session.